I'd like to start off by thanking the organizers for giving me the opportunity to present some of our work. I think my talk's going to be slightly different than everyone else. I'm a signal transduction person who's working her way through species and tissue culture into animal models, and how we are hoping to use metabolic profiling to potentially come up with novel therapeutic strategies to improve clinical outcomes for breast cancer and other cancers. So before I get started, I'd like to thank my group, because I always forget this slide in the end, and none of my trainees are in the audience, so I maybe didn't have to put it here, but I'd like to thank my graduate student who just defended her thesis 2 days ago, so she's no longer a candidate, she's one of us. Good luck for a job. And some of the other graduate students that contributed to the work that I'll be presenting to you, and some of my collaborators around the world and in the US and in Europe. Thank you for their help. So I'm going to give you a very basic slide on signal transduction, the pathway to metabolism. And we're going to start off with the nucleotides. And we know the nucleotides are the building blocks for just about everything. And what I love about the nucleotides, it's conserved across the phyla. So we share similarity with the worms, the plants, the slugs, whales, spiders, cockroaches, and those little pill bugs. So this is what makes us all very similar. And we know that there's some assembly that takes place, and we get the assembly into DNA, and from DNA we get our RNA, and then finally we get our proteins. And because I do signal transduction, for me, for most of my career of 30-something years, I've kind of not paid attention to this end, so I hope not to offend anyone, and I primarily focus on proteins and post-translational modifications to proteins. Sorry. But what we know, what's important here, is to get to this point, there is a significant amount of energy that is required. And the end result is always metabolites. And again, similar to the nucleotides, the metabolites are conserved. So metabolites that are found in plants, although they're more sophisticated than we are when it comes to the metabolic profile, and we share similar nucleotides. And so for me, I now have sort of shifted over this boundary line that I've always had in my head, that although I might be studying proteins, I now need to understand what the metabolic profile is of how my proteins behave in our disease models. And what's very nice is once you know the metabolites, you can reverse engineer and come back and go back and take a look at the DNA that's been damaged or mutated to then give rise to the metabolic profile that we get in our cancers, or in any disease for that matter. But let's start off with the energy sensor. So AMP activating protein kinase is an energy sensor, and Dr. Kempa alluded to it earlier in his talk. And AMP basically is what senses the energy level. AMPK senses the energy level in the cells. And when there's low levels of ATP in the cells, AMPK is primed, and it will actually bind the AMP, which will then set it up for activation by another kinase. In fact, there are two upstream kinases that activate AMPK. The first is CAM kinase, and the second one is LKB1. And both of these kinases will phosphorylate AMPK on a conserved threonine in the T-loop of threonine 172. And once AMPK is activated, it can then elicit its downstream signaling events. But this is tightly regulated, so there always is a balance. The cell hopes to keep this and maintain this balance. And so under starvation mode, the body shouldn't be producing proteins or proliferating, so there would be a conservation of energy. When there's plenty and abundance of nutrients, the AMPK will sense that and will shift the cycle over. So let's look at some of the things that AMPK can do. AMPK is involved in glucose uptake, glycolysis, fatty acid oxidation, mitochondrial biogenesis. And so when any of these are problematic or there's a reduction, AMPK will sense that and shift the ATP to ADP levels in the correct direction. The opposite is true. AMPK will also regulate gluconeogenesis, the production of triglycerides, cholesterol synthesis, fatty acid synthesis, and protein synthesis. And we know there's several drugs that are in clinic today that will help modulate the activity of AMPK. Most people are familiar with metformin and femformin. Metformin is used for the treatment of diabetes for the last 60 years. It's now in various stages of clinical trial for the treatment of solid tumors, because we know if we can regulate AMPK, activate AMPK, then mTOR pathways would be squelched or suppressed. Other activators are resveratrol. And resveratrol, some of you who drink wine know very well that resveratrol is found in the skin of red grapes, so red wine. It's not the wine that's beneficial to you, it's actually the skin. So it's been calculated, some people are smiling here. It's been calculated that you need to drink 7 glasses of wine to see the benefits of resveratrol, but you'll be so drunk that there's just no point. Your liver will be shot, so there's really no point, but you can take a dietary supplement of resveratrol. Another one you may be familiar with is berberine and quercetin. These are also activators of AMPK. Now, another activator of AMPK is the tumor suppressor kinase LKB1. Now, LKB1, there's nothing excitingly different about this kinase from other kinases. It has a traditional conserved kinase domain, an N-terminus with a nuclear localization sequence. It also has a C-terminus that is phosphorylated on various residues by upstream kinases like P90RISC and PKA. It's also phosphorylated in response to DNA damage by ATM. It was about in the 1997, 1998, it was identified as the susceptibility locus for Poitz-Jeghert syndrome, hematomas polyposis syndrome, that I don't know the global statistics, but in Canada it's 1 in 100,000 people present with Poitz-Jeghert syndrome, so it's somewhat rare. Loss of heterozygosity is not necessary. So as you know, with most tumor suppressors, there's a component of loss of heterozygosity. However, with LKB1, a loss of heterozygosity is not necessary, but haploinsufficiency is observed in numerous cancers. And it's also believed that the tumor suppressor function of LKB1 is actually directly linked to its catalytic activity. So if you have mutations anywhere within the kinase domain, you'll disrupt the folding of the ATP binding pocket and you'll lose the catalytic activity of the kinase and cancers will ensue. And in fact, there's been more than 150 germline mutations found in LKB1 that primarily are localized to the regions that encode for the kinase domain. There are also somatic mutations in LKB1. As a matter of fact, in non-small cell lung carcinoma, 30 to 40% of non-small cell lung carcinomas will actually have mutations in LKB1. So if LKB1 is the upstream kinase of AMPK, you can imagine that LKB1 can no longer phosphorylate AMPK, AMPK can no longer negatively regulate mTOR, and potentially this is one of the reasons why the cancers may survive, because mTOR activity is up. And so years ago, and I don't want to compete with mass spec people, I do very basic mass spectrometry in my lab. So years ago, I developed a gel-free method of mass spectrometry where instead of running the gels, we actually immunoprecipitate from cell lysates or tumor lysates. And we went in with a very small concentration, like 2 milligram of lysate. We went in with a monoclonal antibody for LKB1, and then off of the immunoprecipitant, we digested off the beads and we spec'd it. And as a result of that, there's been several targets that we've published on. And the first one that we found was that LKB1 catalytic deficient mutants will actually enhance the expression of cyclin D. And so we found that both the wild type LKB1 and the mutants of LKB1 are recruited to the promoter of cyclin D. These mutants will actually enhance the expression of cyclin D over the wild type. And so we concluded from this paper that LKB1 mutants were oncogenic. And so we've since discovered that LKB1 is actually mutants that are usually found in all these cancers is actually recruited to the promoter of oncogenes, including cyclin D and c-MYC. The next thing that came out of our mass spec study was that we found that LKB1 and its mutants will actually bind to the estrogen receptor and regulate the downstream signaling of the estrogen receptor. And so we also found that the oncogenic mutants will actually inhibit the transactivation of downstream targets of the estrogen receptor. And so we concluded from this study that LKB1 regulates hormone signaling. And so as we're piecing all of this together, it became obvious that these mutants of LKB1 are found in many, many cancers, but also found in many cancers are elevated levels of cyclin D, elevated levels of the estrogen receptor, elevated levels of c-MYC. And so we tried to figure out which cancer pretty much fits this profile. And so we wanted to see if there was a relationship between LKB1 expression and breast cancer, because we've already marked 3 of the 4 hallmarks of various forms of aggressive breast cancer. The one thing that we were missing was their relationship with HER2. And so we scanned a whole bunch of tissue microarrays. And in the first panel, we have the antibody control. In the second panel, LKB1 staining, and of course, the H&E. And what we discovered was that right here, in roughly 55% of all of the cancer TMAs that we scanned, we saw a loss of LKB1 expression. And we don't know if the mutants are present or if it's just loss of expression. But however, if there's loss of expression, there's loss of catalytic activity. We then discovered that 31% of HER2-positive breast cancers actually lack the expression of LKB1. Now, this hadn't been shown before, and it was really quite difficult to convince people. So we had to move from tissue culture type studies and signal transduction to animal models. And so the first thing we did was we crossed the LKB1 flox mouse with an ERB2 Cree mouse. And the resulting progeny, what we found was the ERB2 mice, roughly 50% will develop tumors at 197 days. When we excise the LKB1 gene, we can actually reduce this by 50 days. So the loss of a tumor suppressor actually reduced the latency of tumorigenesis in this model. And so we did, although our tissue microarrays showed a very interesting phenotype, 31% loss of LKB1, we actually now know that when you lose LKB1 in a HER2 background, you can actually drive tumorigenesis. And when we looked at just simple carminalum, it was very difficult to distinguish the type of tumors just from a gross pathology. We couldn't tell the difference between the NIC mice or the LKB1 mice. By Western blot analysis, we confirmed that there's a reduction of LKB1 expression, elevated HER2, and this is just a graphic representation, elevated HER2 in the mice, in the tumors, and reduced LKB1. And so we also know, I mean, these blots would vary. Sometimes there would be no expression whatsoever, but as you all appreciate the heterogeneity of a tumor. And so we were randomly selecting these tumors from the mice, and we couldn't tell just by looking at them which one had more LKB1, which one had less. But ultimately, we can guarantee that by 20 weeks of age, these mice will all present with tumors. And so when we continued with the immunohistochemistry, and this is just a representation of what the tumors look like in one mouse, we see here that in the LKB1 NIC mice, loss of LKB1 expression, elevated ERB2. In the NIC mice, LKB1 expression is still intact. But what we didn't consider until we started doing a lot of immunohistochemistry was that the phosphorylation status of S6 was up. And so S6 is the downstream target of S6 kinase, which is the direct target of mTOR. So when we quantified that, we saw that there was a statistical difference between S6 phosphorylation in the NIC mice versus the LKB1 NIC mice. And so when we basically, if we look at the signaling pathway for the ERB2 mice, we see that ERB2 is up, therefore mTOR is up, and cell proliferation is up. In the LKB1 NIC mice, these mice here, we lose LKB1, you lose the regulation of AMPK. AMPK can no longer negatively regulate mTOR, and everything is up. And this is why the tumors are more aggressive, because they're more aggressive because we're not only tweaking this side of the branch for mTOR activation, but we're also playing around with the receptor tyrosine kinase that regulates mTOR. So we're coming in from two angles. And this is a basic representation of these pathways. So in general, most clinical drugs that are in clinic today are targeting this branch of the mTOR pathway. They're targeting either the receptor tyrosine kinase, PI3 kinase, or why I can't understand why people are attacking AKT, because it's so pivotal. You're never going to shut down anything, even if you go after AMPK. That's my opinion. But so we've got this branch, and this is the branch of mTOR that we work with. We wanted to make sure that it was intact for mTOR, and so we used rapamycin. We took the primary cells, treated with rapamycin, and we can inhibit phosphorylation of S6. But the problem with rapamycin in clinic and why it hasn't been successful, simply because the feedback loops always get turned back on. So over time, you might shut down the tumorigenesis, but eventually the tumors will come back because of these feedback loops. So when we tried to treat with BIBW2992, which is a drug that's in clinic now for lung cancer, we couldn't shut down phosphorylation of S6 or AKT threonine 308 or serine 473, and these are the markers for AKT activity. But when we use TORN1, an AZD AstraZeneca compound 8055, which target both TORC1 and TORC2 of mTOR, we can not only shut down the phosphorylation of S6, but we can also reduce the phosphorylation of 308 and 473, both of which are involved in the feedback loop. And so in our heads, we thought, well, this is a possible strategy for us to actually go in and do some preclinical trials. But before we did that, we wanted to take a look at the mitochondria, because we knew that if we were playing around with or activating or hyperactivating mTOR, potentially we could be hyperactivating the metabolic profile of these tumors. And so just looking at simple transmission electron microscopy, a very old technology, so to try and convince a modern day student to do a technology from before they were born was very difficult, but we're very glad that my student decided to do this, because we could see in the epithelials of the wild-type breast tissue, mitochondria, classic, nice cristae, nice borders, all the membranes are intact. However, in the LKB1 tumor cells, the mitochondria are large, they're engrossed, the cristae are deformed, and when we did the mito tracking, we see that there's a shift to the right. And this shift to the right is suggestive of increased metabolic activity or metabolic mitochondrial content. We then looked at levels of LDH and PDH, and we see in the tumors that the LDH and the PDH levels are up as would be expected. Then we sent our samples off to Genomics to do metabolic profiling by NMR, and we see that the ATP levels are up, lactate is up, fumarate and glycine and a bunch of branched chain amino acids are up, and then the redox potential is reduced. And so we knew that we had not only hyperactive mTOR, but we also had a metabolic mouse in our hands. So then this brought us back to, thank you for introducing Warburg, Otto Warburg, but he was the one who came up with this idea, and Fisher before him, that highly proliferating cells or cancer cells will use lactate slightly differently. And it's surprising that a tumor, a growing cancer, would actually be using the least efficient way to make ATP through glycolysis. And he referred to this as we refer to this as aerobic glycolysis. And so normally, in the presence and absence of oxygen, one molecule of glucose, if it goes through this pathway, oxidative phosphorylation, we end up with 6 moles of molecules of ATP. In the anaerobic glycolysis, we end up with 2 molecules of ATP. In proliferating cells or highly proliferating cells in our body, and with tumor cells, whether there's presence of oxygen or not, the cells will always prefer this pathway, and in the end, one glucose molecule will produce 2 molecules of ATP. And this is highly inefficient. So you're asking, how is this aggressively growing cancer sustaining itself on 2 molecules of ATP? But we now know, since Warburg's first discovery, we now know that the tumor cells are really quite elegant, and there's a potential contribution of energy from the microenvironment, and there's communication between the tumor itself, its microenvironment, shuttling back and forth, oxidative phosphorylation, TCA, and glycolysis, everything working together. But it's going to be very difficult to try and tease out all of these different players. But in our mice, we know we have the Warburg effect because we do see elevated ATP and we see reduced AMPK. And because there's no AMPK activity in our mice, the tumors are highly aggressive. And so we thought, well, everyone in clinic seems to be targeting the PI3 kinase pathway, and so we thought we'd try and do a study where we actually target PI3 kinase, and so we would inhibit the PI3 kinase pathway and inhibit mTOR simultaneously. We know at 20 weeks our mice develop tumors. So at 19 weeks, we would put our mice into an MRI machine, and the MRI machine's not spinning. The mice are just stuck in there, and we would identify the mice with the tumors, and then on week 20, we would actually start them with the 2 drugs, the BEZ-235, which is in various stages of clinical trial, as well as the AZD8055 compound. And we would metronomic dose, so this is low dose every day, and every 7 days, we would place them into the MRI to image. And so here's an example of what the tumors look like from the vehicle-treated mice, what the tumors look like from the BEZ mice, and what the tumors look like from the AZD. And this is just a graphic representation of the data from the MRI analysis. And we can see that There isn't really a statistical difference until day 14, but this is with a vehicle treated, as one would expect. As a matter of fact, the tumors get so large by 21 days that ethically, the ethical endpoints were forced to sacrifice all our studies at 21 days because of the vehicle treatments. With the BES treatment, which is the compound that's in in clinical trials, we didn't see a statistical difference with vehicle compared to vehicle. However, we did see that the AZD compound was more effective at inhibiting tumorigenesis. When we did very simple metabolic analysis using a seahorse, we didn't expect to see a difference in the oxygen use rates, but we did see a difference in the glycolysis. And so compared to vehicle, the AZD compound reduces the glycolysis, whereas the BES doesn't seem to be a difference between, is statistically not as effective as the AZD. When we combine the drugs, we actually see that there is a reduction. However, we believe this is being contributed by the AZD compound and not by the BES compound. And so to move forward, we weren't satisfied with the BES data. And so we wanted to figure out a way, can we actually confirm that glycolysis or metabolism was involved? And the best way to do that was to use a compound, the 2DG that we heard about earlier. And so same setup, we identify mice with tumors at 19 weeks. At 20 weeks, we start the treatment. And this way, by inhibiting mTOR and glycolysis, we were very curious to see what would happen to the tumors. And so on day 0, these are the different mice in the MRI, and the yellow circles are highlight where the tumors are, and there's a small tumor here, tumor, and tumor. And so these are the different treatments: vehicle, 2DG, AZD, AZD, 2DG. And we can see that on day 21 of vehicle, not only did the tumors get bigger, but they were more tumors. With the 2DG, we do see some tumor growth, tumorigenesis. With the AZD, we see a modest increase. It's when we combine the two drugs that we actually go from this to this. There was a reduction in the tumorigenesis. And so here again, we see vehicle, 2DG, AZD and 2DG combined. We see that the combination itself was the most effective at inhibiting tumor growth. And not only did it inhibit tumor growth, but it also inhibited tumor burden. So those of you who work with mice, you know very well they have the potential to actually have 10 tumors. There's 10 teats, 10 tumors, right? So in vehicle, we pretty much, every mouse would have up to 10 tumors, and sometimes the tumors would have tumors, so it could be up to 15 tumors per mouse. 2DG, there were still tumors, but by the time we got to the combination, we reduced the tumor burden to 1 or 2 tumors, and they were always significantly smaller. When we started to interrogate the signaling pathways, we see that what's the most interesting thing with the AZD, we know that it will inhibit the phosphorylation of S6, but what we do see is a expression of the phospho-ERK and the P90 risk. Now these are the pro-survival pathway loops that get turned back on. If you inhibit mTOR for prolonged periods of time, as we know in clinic with rapamycin, the feedback loops come back. So the ERK pathways, ERK gets phosphorylated and activated, and P90 risk gets activated. With the 2DG, we see that the S6 is still present, is still phosphorylated and active. With the combination, though, we see a slight reduction in phosphorylation of S6, but we do not see the feedback loops. The feedback loops have been prevented. And so we believe that what we're observing in the preclinical trial with the mice is because the 2DG combination with AZD turns off the feedback loops. Now what's interesting, if we look at the pathway, so if we treat with the AZD 2DG, we're turning off mTOR, turning off glycolysis, and we're also shutting down this feedback loop. Now, what's interesting, if you ask people who do the studies with the BES compound, for example, and you ask them if they've gone back to the patients to see if the pro-survival pathways have been turned on, not a single clinical study at this time has actually gone back to look at the pro-survival pathways. So I would gamble to say that eventually those tumors will come back in the women who are currently in trial for these drugs. So as to finish up, what we're doing now is we're taking all of these tumors from our drugs, and we've done metabolome-wide analysis, and we're doing genome-wide association studies. And of course, my lab doesn't do this, so we end up sending everything out. And Metabolon has identified 65 interesting metabolites out of their 600 and something that might be very interesting for us to pursue. And in the meantime, we're in the process of doing the genome-wide, and we hope to be able to find some sort of correlation between the metabolites that we've identified and something with the genome information, and eventually work back up the pathway to figure out what signaling or what genes have been affected in these tumors. And quickly, we've learned we've got a mouse model now that we can interrogate metabolic processes. We believe that LKB1 should be considered a metabolic marker for breast cancer. So if a woman comes into clinic and has reduced expression of LKB1 or reduced phosphorylation of ACC, which is a marker also for AMPK, then we really need to think about what sort of treatments to give this person, because mTOR is definitely upregulated. We want to leverage what we've learned with our breast model, and now we have other animal models, other cancers and neurological disease models. We're pursuing other clinical trials, and we're developing small molecules to target metabolic processes. And with that, I'd like to thank you, and any questions. Yes?
So a quick question I would have is, have you ever tried to do these experiments changing the oxygen conditions, such as hypoxic or anoxic or super high concentration of oxygen conditions?
Well, you have to keep in mind, these are in vivo studies, right? So we know with some of the tumors, they're necrotic when we open them because there is a component of hypoxia happening. But because this is, it's not tissue culture, we really can't ethically to put an animal in one of these metabolic chambers, and they already have cancer. So I can tell you in Canada I might have a problem getting that past animal ethics to give them cancer and then try and take oxygen away from them. Right. But in tissue culture, it's a perfect experiment for tissue culture, and that's a great idea for us to do when we're doing our seahorse analysis, metabolic stuff with the seahorse. But with the animals, keep in mind these tumors are already going through various stages of hypoxia, anoxia, and we can tell when we open them up which tumors, and this took us a long time to figure out, the tumors that we would go after because they're big and juicy are not necessarily the tumors that give us the best primary cells or data, right? Because everything's dead. Autophagy, everything's dead. But for tissue culture, taking primary cells and putting them in culture and doing quick studies is perfect. However, the caveat to that is we discovered very quickly that when we take primary cells from the tumors or from wild-type tissue and place them into culture, within a half hour, 30 minutes to 40 minutes, the metabolic profile already changes because of the media. And so you have to be very cautious when you're working with primary cells from patients or from your animal models. The minute you stick them into tissue culture, you have to be very conscientious of how much glucose is sitting in your media, because you will change everything. Okay?
Okay, and one other question. If you were to give somebody a monoclonal antibody like Herceptin, would that increase the amount of LKB1 that you would produce?
Well, we have looked at that. And so the expression level, at the gene level and at the protein level, proteins is where it's at, actually doesn't change when we give the trastuzumab. The gene level and the protein levels for the kinase do not change. But we do know we lose ERB2 levels when we treat the animals with the trastuzumab. Okay? But good question. Thank you.